This is the RNA World Hypothesis, proposed over 40 years ago. While studying exoplanet 21, we found it to be very similar to planet Earth when it was 4.2 billion years old. The planet was very volcanically active during this time, and the chemistry of the Earth was producing random chains of RNA. These chains of RNA formed in a sort of primordial soup where nucleotides were present. Some of these chains were stronger than others, and many degraded quickly. This early RNA was made up of 4 base pairs and a backbone known as ribose, which we'll get to later.
With the advent of RNA, there was a lot of competition for survival in this soupy world. This competition allowed for more useful and better strands of RNA to survive, and eventually, RNA was able to replicate.
RNA strands found ways to copy themselves via base pairing, but mistakes in this led to mutations in sequences. These mutations allowed for RNA strands to be better or worse off in their environment.
Replication was all based around those base pairs we mentioned earlier.
The 4 base pairs present in RNA are adenine, uracil, cytosine, and guanine.
These 4 base pairs or nucleotides are connected by the backbone to one another, allowing for diverse sequences of RNA. During a study, researchers found that one strand of RNA allows free-floating nucleotides to bond with it, forming a complementary strand. When the water the RNA was in was heated, the base pairs lost grip and were split apart.
The 2 strands then had more free-floating nucleotides attached to them, making 4 strands of RNA from one original strand. RNA strands are also able to fold into complex shapes and base pair with themselves when there's a lack of nucleotides present. Certain bases are unable to find a pair and thus point outward. These bases cause unique chemical reactions with their surroundings. These special RNAs are called ribozymes. Ribozymes have varying functions depending on their shapes and their sequences. Over time, DNA and proteins evolved from these constantly changing ribozymes and RNA. This DNA was superior and took over for storage of genetic information.
Due to mutations previously mentioned, RNA was able to diversify into a plethora of things. This development of RNA is still going on today, within each and every one of you. Today, RNA is able to slice, dice, catalyze, code, build, destroy, replicate, and transform. Although this theory has a lot of backing to it, it's not perfect. The main issue researchers had was that the backbone of RNA would not bind in the wild due to a lack of enzymes. In their experiments, researchers had to do this themselves.